Alright guys, welcome to Pathophysiology. What we're going to do in this lecture is we are going to do an introduction to what patho is, and then we're going to do a little bit of reviewing from chapter 1 where we're going to look at reviewing cells and transport and things like that that you should have already gotten back in biology and even in anatomy 1. So what is pathophysiology? Well, it's based on common or classic presentations of a disease. Now, this is in the physiological functioning of the human being. So in other words, it's the study of what happens when normal anatomy and physiology go wrong. So when things are not working like they should, that's when we're studying pathophysiology. Now, there are some interrelated topics that go along with pathophysiology. We see that there is etiology. This is going to be the study of what causes a disease. Pathogenesis is the organization and how the disease progresses. We have clinical manifestations. This includes things like signs and symptoms. So these are what the patient kind of presents with when they have a certain kind of issue. And then also the treatment implications. How do we treat when these things go wrong? All right, so these are some related things. And when we go through each of these chapters in Pathophys, we're going to first look at, okay, what is the abnormal, like what went wrong, how, like why did it go wrong, what's kind of its progression, and how do we treat it. And we'll be looking at that in every chapter as we go through this course. So before we get to the point of where we talk about disease, we need to understand what health is. So when we say that somebody is healthy, we mean that they have an absence of a disease. So what is a disease though? Because it just says the absence of, meaning it's not there. So what is a disease? A disease is a state when bodily function is no longer occurring normally. So when things are not working like they should, or what our kind of status quo is, then we see that we have something that we would consider a potential disease. Now this is also known as an alternation of homeostasis. So remember, if you'll recall, homeostasis, homo means same, stasis means status. So this is the same status. And you have a lot of stuff in your body that is supposed to stay at a certain status. You have things like body temperature, blood glucose levels, amount of water that you have in your body so you're not dehydrated. There's a lot of things that you are not even actually consciously aware that your body is constantly monitoring. And if any of those get out of whack, we want to put it back to homeostasis. If we're unable to do that and we're not able to get back to homeostasis, then we have a problem. All right, and so this would be what we would call a disease. Now, diseases can be classified in a number of ways. I have a list here, and there's a lot of them that they can fall into multiple categories. All right, so we could have a congenital metabolic disease. There could be a genetic inflammatory disease. So it's not necessarily going to fit into just one of these classifications. It might fit into multiple. So let's look at what these classifications actually mean. All right, so when we look at a disease, if we say it is hereditary, this means that it came from your parents. It was passed down from your parents to you. So this does have a genetic basis, which means that it also would fall into what we would consider a genetic disorder as well. We also see there's congenital. Congenital means that it was present at birth. It was there. We may have detected it or we may have not detected it, but it was present from birth. We also see genetic. Genetic just means that the genes are involved. Now, this could mean it's passed down from your parents, but it could also mean that it was a spontaneous genetic mutation. So genetic and hereditary do not necessarily mean the same thing. All hereditary diseases are genetic, but not all genetic diseases are hereditary. All right, and so we do see that there is a little bit of a distinction there. We also see that there's some developmental diseases. When we look at developmental, this means that it does cause effects on the development of an individual. It could mean it could affect their it could affect their walking ability, like learning how to walk, learning how to crawl, learning how to sit up. It could affect their talking. There's a lot of different things developmentally that we see could be affected. We see inflammatory. Inflammatory is related to the inflammation process, and inflammation has a couple of cardinal signs that inflammation is taking place. It's normally swelling, redness, heat, and pain in kind of that area. So when we look at that, it could be due to inflammation. We also see that there are degenerative diseases. Degenerative means it's progressive. Okay, there's a deterioration that happens, and a lot of times this could be leading to a loss of function, but sometimes it's due to being like overly used. We see a lot of like degenerative diseases, like when we talk about certain types of arthritis. We've used that joint so much that it's broken that joint down and we lose that function. We also see that there are metabolic diseases. Metabolic diseases are, of course, going to affect metabolism. And if you'll recall, metabolism is the sum of all the chemical reactions in your body, which means if we're looking at a metabolic issue, a lot of times it deals with like enzymes, things like that. We also have neoplastic. Neoplastic is where we're looking at cancer. Neoplastic disorders are where there's a mutation in the genetics that causes those cells to kind of go rogue, and those cells start to divide uncontrollably, potentially causing cancer. All right, so that's what we're looking at. And so when we look at these diseases, when we talk about each of these throughout the semester, they're going to fall into at least one of these categories, and sometimes multiple categories. Okay, so before we move on, let's look at homeostasis in a little more detail. So what is homeostasis? Homeostasis is an equilibrium, a balance, a consistency, or a stability. So if you'll notice, all these words kind of mean the same thing, but the whole point is that it keeps your body in a kind of constant state when we're looking at homeostasis for you. It's a self-regulating, give and take system. So it's going to regulate itself. When it gets out of whack, it's going to try to put it back to normal. We also see that it responds to even minor or minute changes in your body, and it compensates for them through different mechanisms. So your body's going to have ways to help compensate and put you back to normal if they're working properly. So there are two types of feedbacks. There's two types of processes that can restore homeostasis. The first is a negative feedback. When we look at a negative feedback, guys, it's very similar to what you see in the picture here. And this picture is from your textbook. This is kind of like how your thermostat works in your house. You set it at a certain temperature. Okay, let's say you have it set at 72. If it gets too high, it goes above 72. Okay, so we have it set and it gets out of whack, it goes too high. We see that the air conditioner kicks on and it stays on until it restores it back to 72. Then it shuts off. Okay, and then on the other hand, if it gets below 72, your heater kicks on and it brings it back up. So there's this constant kind of give and take, and it's adjusting based on what's happening right then and there. This is how negative feedbacks work in your body as well. So when we look at a negative feedback, let's just say that it deals with like water balance. You are now becoming dehydrated. You don't have enough water. There's a number of things that'll start to happen in your body. Your kidneys will start to try to compensate with that, and they will hold on to as much water as possible. Okay, making it where you create less urine. Also, you want to replace the water as well through drinking, so you might start to get thirsty and where you want to drink something, and so we want to restore it back to normal. All right, other times when you look at blood glucose, hormones play a big role in that. Insulin, when your blood glucose gets too high, insulin gets released. It tells the cells to absorb the glucose out of the blood, restoring the level back to normal. So these are most of the systems in your body that do homeostasis are what we consider negative feedbacks. On the other hand, there's also positive feedbacks. When we look at positive feedbacks, they're not necessarily positive for the individual. Okay, it's kind of like misnamed in a way, but when we get out of whack, it actually makes it worse before it makes it better. Okay, and there's a couple of situations where this occurs. Now, males don't have as many of these. They do have a few, like fever. A lot of times, sometimes our fever will get worse before it gets better, and that's to try to help fight against the invader that's come in. We also see with bleeding. A lot of times when we look at bleeding, we want to bring more blood to the area so that more platelets and all get there to clog up the vein or artery or whatever has been broken, the vessel that's been broken. And so the bleeding sometimes can get worse before it clots and gets better. But females have a lot more of these. Females, anytime we deal with like childbirth, breastfeeding, all of those are positive feedbacks. So an example with childbirth is when it's time to have the baby's head presses on the cervix, stretching out the uterus sends a message to the brain releases oxytocin, it sends it down and it tells the uterus to contract to help push the baby out. When the baby's not pushed out yet, it sends the message back up, it releases more oxytocin, and it continues. So contractions start out kind of mild, further apart, and they get more extreme, closer together, and all that stuff until the baby's born. The thing is, though, once the baby's born, those contractions don't just stop. Okay, it has to go back to that other progression where they start to get further apart and they start to spread back out. Okay, until they finally go away. And so we see positive feedbacks a lot of times can be very vicious cycles in our body to hopefully get us back to normal. All right, so we're going to look at some of those subcategories we talked about that deal with pathophys. And so one of these is what we call etiology. This is the study of causes or reasons why we get diseases or certain phenomena, okay, in our bodies. The whole point of this subcategory is to identify what factors caused the issue. Okay, are they acting with multiple things to provoke a disease or an injury? Because sometimes it's not just necessarily one thing that's causing your problem. It could be multiple things. So when we look here, you see here we have like a little flow chart. Let's put up here, we have etiological agents. Some of these agents could be biological. When we look at the cause of a disease that's biological, that could be a bacterial infection. That could be a viral infection. It could be, you could have worms, a worm infection like tapeworms or something like that. So this is going to have a biological component that deals with that. On the other hand, we also see that there's a chemical, there could be a chemical component. Chemical components could be that you have been exposed to certain chemicals that cause burns, like if you get acid spilt on you or something like that. Other chemical components could also affect you inside. So if you ingest something that you shouldn't, it could cause chemical issues. We also see physical issues. Physical is going to deal mostly like with trauma. All right. So trauma would be something like a car accident, a gunshot wound, other kind of burns like through fire, not through chemicals, but through fire. On the other hand, we also have nutritional issues. These nutritional issues could be excessiveness, where you get too much of something which causes an issue, or it could be defects or deficiencies where you don't get enough of something. So this could be a deficiency where you're not getting enough of something. So when we look at this, if you have an iron deficiency, you don't have enough iron, it can affect your blood levels and it can cause you to be anemic. On the other hand, we could have an excessive amount of, say, like iron or something else that causes you to have a toxic level. So there's a lot of different things that can affect us even nutritionally when we look at this. So when we look at etiological causes, we also want to be able to classify them into some different categories. So some classifications that we look at here at first are the first one is idiopathic. If it's idiopathic, the cause is unknown. We don't really, we've done our investigations, we've done a lot of tests, and we do not know why that individual has dealt with that issue. All right, and so when we look at it being idiopathic, the cause is pretty much unknown. We see there's iatrogenic, and this one is where we see the cause results from unintended or unwanted medical treatment. And so this happens sometimes whenever we are treating for one thing and it causes another disease or issue. An example of this would be something like with chemotherapy. A lot of times you have to have the chemotherapy to take care of the cancer, but it compromises your immune system to where then you can end up having other types of diseases that come in. All right, and so that's what we're looking at here. Another thing is we want to look at risk factors. A lot of times an individual might be at risk at developing a certain type of disease due to certain factors that are present in their life. There's a lot of different types of risk factors for different diseases. It could be based on your race or your ethnicity. It could be based on your gender, whether you're male or female. It could be based on your age. Some individuals at a younger age are going to be more prone to certain things versus at an older age. Now, this does not mean that you definitely will get it when you're younger or older, or you definitely won't later on. It just means that you're at a higher risk at that particular point. So there's a lot of different things that can be considered risk factors, whether you smoke, drink, you're overweight. Again, risk factors can be pretty much any kind of thing that deals with the individual. All right, so when we look at pathogenesis is the development or evolution of the disease. Now, evolution just means change over time. So it's how the disease progresses or changes as we do certain kinds of treatment, certain types of medications, things like that. This is from the initial stimulus to ultimate expression and manifestations of the disease. So a lot of times when you first start getting that cold, you might feel a little stuffy and a little bit and feel not too great, but then it can continue to progress to where then you can't get out of bed and your fever's super high and you have all these aches, and there's a lot of things that could happen. And so that development is what we call pathogenesis. Now, the description of how the etiological factors are thought to alter your function and lead to the development of those signs and symptoms for a particular disease or disorder is what we term as pathogenesis. All right, so somebody who studies pathology, they're wanting to see that progression of when it started until it finally manifests to where we know what it is. All right, so that's what we're looking at with pathogenesis. So let's look at what a clinical manifestation is. Now there's two types of clinical manifestations. There's signs and symptoms, and there is a difference. You do need to know the difference. When we look at a sign is an objective or observed manifestation of a disease. So this is something that you actually observed or you can see. All right, so this would be like if you take blood pressure, you can actually get the blood pressure. If you see them vomiting, okay, that is obviously then a sign because you saw it, you observed it. On the other hand, we see symptoms. Symptoms are subjective feelings, okay? This is where the patient tells you how they feel, all right? So when you ask them what their pain level is, that's going to be subjective, and it's going to be different for each person. If they come in and say that they've had a fever and they were vomiting, that's subjective until you have observed it or seen it. Okay, so we would say it's more of a symptom unless we see it. If we take their temperature and it's high, then becomes a sign. All right, so there's the difference between a sign and a symptom. All right. On the other hand, we also see that there are what we call syndromes. This is the etiology of signs and symptoms that has not yet been determined. And so we see that a lot of times when we say that they have a certain kind of syndrome, like Down syndrome, there's a whole list of things that individuals with Down syndrome may deal with. Now, that one individual may not have all of those things that are going on, all right, and they may not have all of them, but it gives us a list of things that we can see that might come along with a certain disease the more and more that we study it, all right, and so this is going to be something as you study different things, they may get a name of a syndrome if we start to see a lot of different signs and symptoms that come into play. There are certain stages that the patient kind of goes through. All right, and so when we look here, the first stage or phase is called the latent period. This is the time between when you were exposed, your tissue was exposed to an injurious agent, or it's the first appearance of your sign or your symptom is what we call the latent period. All right, and so you've been exposed. You've been exposed and it may take several days before you actually start feeling bad. Okay, so this is what we call the latent period. We then have the prodromal period. This is the time during which signs and symptoms appear indicating the onset of disease. So we go from latent to what we call the prodromal period. So this is where you've been exposed. Say you got exposed to the flu. Okay, somebody touched the door handle, then you touched the door handle, now you've been exposed to the flu. You are in the latent period. Now, when you actually start experiencing the signs and symptoms of a flu, you start to get the body aches, you start to have the stuffiness, you start to have the fever. Now you've moved into the prodromal period. We also see that there's what we call the acute phase. This is the disease or illness reaches its full intensity. It's gotten to its kind of apex, the top amount. This is normally when you feel the worst. Normally after that point, then hopefully you start to feel better, but this is what we call the acute phase. We also see that there's what we call the acute clinical course. This is if the disease or issue is short-lived. It may have some severe manifestations, like you really feel terrible. It could even be life-threatening, but it is a short-lived disease, meaning that it apexes really quick and then we start to see improvement normally after that, if a lot of times if treatment in those real life-threatening cases is given. On the other hand, there's what we call chronic clinical course. So when we look at chronic, this may last for months to years. It sometimes then is followed by an acute course, but chronic means that it's like long term. This is something that you deal with for a long time. Some of it examples of like a chronic clinical course would be like rheumatoid arthritis, some sort of autoimmune disease like lupus. It's something that you kind of live with for a very long time. Now, there's also going to be some different things that take place, especially when we look at like a chronic disease like MS or lupus or rheumatoid arthritis. There's going to be times where the patient may deal with what we call exacerbations. Exacerbations are a sudden increase in the severity of the disease or the signs and symptoms. And so we see that they were kind of at the status quo and then they started feeling really bad, like we start to see major issues. And then it may come back down, but that's an exacerbation. On the other hand, there's what we call remissions. Remissions is where there's a decrease in the severity of the signs and symptoms. This may indicate that the disease is cured, but it may not. So a lot of times you hear about like cancer patients when they say I'm in remission. That just means that we can't detect any of the cancer anymore. They may be cured, but it could come back. Okay, and it could come back in what we call the exacerbation. On the other hand, we see there's convalescence. This is a stage of recovery after the disease, injury, or surgical procedure. And so even though we have taken care of the underlying disease and issue, there may be weakness that's part of that. There may be some things like physical therapy that may need to be done to try to get you back to completely normal. It's a recovery stage. And then we also see that we have what we call sequela. This is subsequent pathologic condition resulting from an illness. So this is something that you had the illness and the issue and it triggered something else. Okay, so you had the cancer, you got the treatment, the cancer is kind of under control, but because of that treatment, it triggered something else where you developed like severe pneumonia. Okay, and so when we look at that, it can trigger it. The same thing kind of happens like when you have a viral infection, all right, and you're not feeling really good, and you go to the doctor, and then the doctor gives you antibiotics for a secondary bacterial infection. Your body was so busy fighting off the virus that then a bacteria infection came in as well. So this is something that happened because of the illness that you had at first. All right, so that's what we're kind of looking at here with these terms. All right, so there are some concepts of normality and health and disease, and these can deal with like individual factors. When we look at an individual, there are some cultural considerations we need to look at when we are treating an individual. Each culture defines health and illness in a manner that reflects their experience, and so in some cultures, they don't send people to the doctor very often. You have to be very severely ill before they'll take you to a doctor. In others, in some cases, they don't believe in modern medicine and they want to use more of their traditional type stuff. And you need to respect that as a medical professional. It could be due to a religious issue. It could be due to, again, their culture. And you need to respect that if that is a decision that they have made. You want to offer them the options of what can be helpful to them, what the modern medicine can do for them, but ultimately it is the patient's decision. Also, there's some individual factors like age. A normal value for one person at one age may not be normal for another, and we're going to actually see this like when we look at body temperature, when we look at heart rates. They're going to be different based on their age. With kids, a lot of times we see that if their body temperature gets to a certain level for fever, it's a lot more dangerous than it is for an adult. We also see in older individuals, their normal body temperature is normally a little bit lower, and so a fever for them maybe like 99 or 98, which is not really a fever for anybody else. And so when we look at that, you have to look at age differences. And then we also have to look at gender differences. The normal value for a man is not going to be necessarily the same for a woman because body size is different, hormones are different, progressions of diseases can take place differently in a male or female. And so it's really important to look at all of these differences and take those into account when we are trying to do kind of diagnosis and treatment. There's situational differences that determine whether a deviation or from normal should be considered abnormal or an adaptation mechanism. Sometimes when we are exposed to different things, our body adapts to them, and so it creates a new normal, and that's not necessarily bad. That's what adaptation does, and so we need to look into those different situations to see if that was potentially the cause of the issue. There's also time variations that need to be taken into account. The time of the day or night, the time of their sleep rhythms and different things like that could also affect how the individual is feeling. One example of this, and we'll talk a little bit about it later on in a chapter, is Alzheimer's disease. Sometimes there's certain times of the day that person may be better than others. And so we'll take a look at how there are variations in time that can affect an individual as well. Now, once we've determined or we think we know what's caused the disease, we've seen the signs and symptoms, then we need to go into the point of diagnosis. We need to identify and name the actual disease. Okay, so now we know what caused it, we know the signs and symptoms, now we want to classify it into a certain name. This is really important because you need the history of the patient and that physical exam in order to determine exactly what they may have, and diagnosis helps lead us to treatment. Once we know what they got going on, we can do treatment, which are strategies used to manage or cure the disease. We also, once we learn about certain diseases and we continue to study them, we can work on strategies to prevent them, and this is what we call prevention. Some preventative measures are things like going and getting certain kind of checkups regularly, like pap smears for females, or mammograms, or colonoscopies. Those all can be things that can help with prevention when we look at this. Prognosis. Prognosis is the individual's likelihood to a full recovery or regaining normal function. In some cases, the prognosis is not good on getting normal function back. They may live and survive, but they may not get complete function back in certain organs or body parts or things like that. But that's the prognosis. Now, when all this is said and done with treatments and different things like that, there are things that we call complications. Complications are new problems that arise because of the disease. They could even be what arises based on the treatments of that particular disease. All right, the last kind of thing we want to look at when we're looking at this introduction to patho is epidemiology. Epidemiology is where we study the patterns of diseases in a group of people. And when we look at epidemiology, it's important because we want to see kind of trends that might be happening. It helps us determine what vaccines need to be maybe made available, like with the flu vaccine. They watch the trends and the patterns of the flu, and then they try to determine which ones are going to be the issue, and they package those in the flu shot. And so there's this whole part is very important. Individuals who really study epidemiology, this would be something like the CDC. People at the CDC go through this. WHO, which is the World Health Organization, individuals that work with that. And so they're watching to see if there's certain patterns to be able to warn us if epidemics are happening, or if we find out that certain organisms carry a disease, like we found mosquitoes carry malaria, things like that. This is what's important with epidemiology. Another thing we want to look at too is levels of prevention. Once we know a lot about a certain disease and its patterns, we can try to offer suggestions on how to prevent issues. So when we look here, there is a level that we call primary prevention. Primary prevention is where you do not have the disease, but you're trying to prevent yourself from getting it. And so one of the best examples of this is vaccinations. All right. A lot of you probably haven't had measles or mumps. And to be honest, once the chickenpox vaccine was given out, a lot of individuals haven't even had the chickenpox. When I was a kid, the way that you got the vaccination for chickenpox was go play with somebody who has chickenpox and you get it, okay, and your body fights it. Nowadays, you don't have to do that due to vaccinations. On the other hand, there's another level which is called the secondary prevention, and this is where your disease may be detected. So these are going to be things that we do to try to detect if the disease is there, but we want to try to detect it early. The earlier we detect something, the easier it is going to be to treat it. So an example of this would be like pap smears for females, because used to that would be yearly, but now with genetic testing, they've actually expanded that to where you don't have to get them as often because we know what the precursors look like. Yearly physicals are also important, so they can watch like your blood pressure, your heart rate, things like that. And so all those things can be taken into consideration to hopefully catch potential issues early. That's the secondary level. Now, the third level is where you've already gotten the disease, okay? You've already gotten the problem, but now we're going to try to prevent the problem from coming back or causing more issues. And this is what we would call rehabilitation. So let's just say you already had the issues with your heart. You've had open heart surgery. Now you're going to go to cardiac rehab, and that's to help your heart rehab, and we change your diet, and we try to get you exercising, and all those different things to help prevent it from coming back again, preventing it from being an issue again. And that's the tertiary level. Okay, so we have primary, which is first, secondary, and tertiary, which is third. All right, so now going back to epidemiology real quick, when we look at epidemiology, we are studying the patterns of disease, but we also are going to examine their occurrences, their incidence, how prevalent they are in certain areas, how are they transmitted, the distribution of diseases in large groups of people. Like, do we find that it's going to be with people who travel a lot? Did one person bring it from this area to another area? There's a lot of different things that we would look at for epidemiology. Now, when we study epidemiology, there are going to be some different types of diseases that come into play. And so the first is what we call an endemic disease, and this is a disease that's native to a local region. Okay, so an endemic disease would be something where it kind of affects that region locally and it's an issue, but in other places in the world it's not a problem. Okay. Endemic diseases, though, can spread to what we call epidemic diseases. Epidemic diseases spread to many people at the same time. All right, and so this is where it might be in a local area, but lots of people are exposed to it and are sick with it, or even it could spread to other areas as well. And then we see pandemic diseases, which means that it's spread even further to large geographic areas. So it's a progression. It starts as endemic, it becomes an epidemic, and then it can become pandemic. And so when we look at this progression, we want to stop it before it gets to this whole pandemic side. And so in the news, a lot of times if you hear of like somebody that, like when the Ebola recently was a big deal in Africa, and that individual who worked over there and he came over to the United States, he kind of lied about stuff, but he had Ebola. He potentially would have caused a pandemic. He could have caused the spread of the disease to go to other places. And if multiple people do that and they travel to other areas without receiving that treatment, then it potentially could cause major issues. And this is a lot bigger issue nowadays than it used to be because of how free it is to travel. Like with flying, you can fly from one place in the world to another within hours, whereas before you'd have to get on a boat and it would take days. All right, and so the spread of disease can happen a lot quicker nowadays because of our transportation means that we have. Now, things that can affect the progression of the disease or variables that can come into play are age. Sometimes certain individuals, due to their age, are going to again be more prone to contracting the disease. Ethnic groups, certain ethnic groups have a higher chance of being exposed. Gender, again, male or female, and a lot of times this is based on hormones. Socioeconomic factors and lifestyle considerations can also affect this, and also geographic location. Obviously, if there is a breakout of something in Africa, we over here in Hobbs or in Lee County don't have to worry so much about it unless it travels here. Okay, so like with West Nile virus, it used to not be that big a deal because it was over there west of the Nile in Africa. However, now we actually have seen that there are some cases of West Nile that have been in Lee County. Now we're going to have to worry about that a little bit more. And so when we look at this, geographic location could play a role, but then we also can watch to see if the spread of a certain disease has happened due to this geographic location. Right? So this covers the kind of introduction part of pathophys, and so now we're going to move into the chapter 1. Now when we look at chapter 1, this first part of chapter 1 is just review. I'm going to briefly review some different things about cells and organelles and things like that, so that way when we come to class we can focus solely on the pathophys part. We can dive deep into the newer stuff that you're going to be learning in this course. All right, now you're still going to be held accountable for this presentation, the introduction and the beginning of chapter 1. It's still going to be tested on, okay, but you will view this outside of class. All right, so when we're looking at chapter 1 in pathophys, the first thing you want to look at is what is a cell. Now, cells are the building blocks of life, and they work together to form tissues. Tissues work together to form organs, and organs work together to form organ systems. So guys, even at the cell level, if the cell is abnormal and it's not working properly, it can affect the tissue, which then affects the organ, which then affects the organ system, which then affects you. So the cell is our kind of basis here when we're looking at this. And so we're going to be looking at some cellular abnormalities, like when the cell is the issue. We're going to look at tissues, organs, things like that throughout this semester. So when we look at the cell, the basic cell functions, there's a couple of structures that are super important in the cell. Now the first, of course, is the cytoplasm, also known as protoplasm or a cytosol. This is a colorless liquid. It contains mostly water. However, it also has nutrients, ions, dissolved gases like carbon dioxide and oxygen, and even waste products that the cell has produced in this cytoplasm. Now, this is where all of cellular work takes place. All the little internal structures in your cells are going to be in the cytoplasm, and they're going to be doing their work. Okay, so this is the location where all the work is occurring. Now, who does the work? The work is done by organelles. Organelles are little organs, and they perform the work that maintains the cell's life. Now, in your textbook at table 1-1, it lists all these organelles for you, and you do need to review them. I'm going to review them briefly here in the PowerPoint, but you do need to take a look at them in your textbook as well as take a look at the diagram of the cell. So when we look here, the nucleus is also a very important structure. The nucleus is the control center of your cell. It regulates your cell growth, your metabolism, and your reproduction. It's kind of like the brain or manager of the cell. And now the reason that it manages everything is it contains your genetic information, your DNA. Now if you'll recall, the DNA is made out of 4 base pairs. These 4 base pairs are A, T, G, and C. So if you remember, A is adenine, T is thymine, C is cytosine, and G is guanine. All right, so you need to make sure that you know that these 4 pairs. Also recall that A pairs with T and G pairs with C. All right, so if you look here, here is the structure of the cell. You can see the different organelles who are labeled. You can see here the nucleus in the purple. We also see that you have the cytoplasm, which is the liquid portion here. We also have the plasma membrane, which covers the whole outside of the cell. Now within the cell with these different organelles, you see things like the mitochondria, which is a bean-shaped structure. This is the one that does is the powerhouse of the cell. It creates energy. We also have the lysosomes. The lysosomes are going to clean up the area. They're the recycling center. Right around the nucleus, you're going to see that we have the endoplasmic reticulum. There's 2 types. There's rough and smooth. Rough ER is going to be studied with ribosomes, who are important in protein synthesis, and so the rough ER is going to help with protein synthesis, whereas the smooth ER is going to lack ribosomes, which means they're going to help produce lipids and also detoxify the cell. Over here, this kind of stack of pancake structure is your Golgi apparatus. It's like the UPS store. It's going to package things up and send it out of the cell, which you can see here. All right, so we have a lot of stuff that goes on within the cell, and each of these organelles have to do their job in order for your cells to be successful. So let's start with the plasma membrane. If you'll recall, the plasma or cell membrane is a semi-permeable membrane, which means that it only is going to let certain things come through. It surrounds the outer part of the cell. It's also made of a phospholipid bilayer. So if you look here, the phospholipids, the head area is hydrophilic, meaning it likes water. The blue tails here are hydrophobic, which means that they don't like water, and they're made of the fat. And so this creates a barrier or layer. So outside of the cell has water, inside of the cell has water, but this middle layer here between those two do not. All right. We also see there's proteins embedded throughout the membrane, which allow things to pass through it. But again, it's selectively permeable. It's only going to let certain things come in and out of the cell. All right, so now let's talk about these little organelles or these functional components. So the first one we want to look at are the ribosomes. The ribosomes, guys, if you recall, are the site of protein synthesis. Protein synthesis means we're going to make proteins. These guys are the ones who are going to create that amino acid chain, which is the first stage of the protein. These may be attached to the rough ER, which is the ER stands for endoplasmic reticulum, not ER here, or the ribosomes could just be freely floating in the cytoplasm. The endoplasmic reticulum is a matrix of paired membranes and vesicles. It has some tubular connection work or systems, so it connects. It's the place where metabolic activities can occur, like making, again, proteins if it's the rough ER, or lipids if it's the smooth ER. So the rough ER is going to produce proteins for the membranes and lysosomal enzymes. Okay, and you can see it here. It's rough because it has the ribosomes on it. On the other hand, the smooth ER is going to create lipids, which are fats or lipoproteins, as well as steroids, which all are fat related. It's also going to regulate your internal calcium levels. It's going to help with metabolism, and it's going to help detoxify. It's going to help detoxify certain hormones and drugs that come into your body. The next is the Golgi apparatus. Remember, this is kind of like that stack of pancakes I talked about. It's also known as the Golgi body. As long as the Golgi is there, you're good. This receives or processes and also transports newly synthesized proteins or lipids. So again, like I said, it's like the center who is going to, like the UPS store. We don't really have one of those here in Hobbs, but if you've seen the commercials, the UPS store is, you can come in with anything you want. Like I can say, hey, I want to ship this. It comes in, they put it in the box for you, they package it up, they put the label on it, and they send it out for you. This is what the Golgi does for your cell. Now, the lysosomes, on the other hand, these guys are going to be kind of like the waste management of the cell. They're going to break down the cell products and the foreign bodies so that they can potentially be used again. So they're going to recycle what they can. The issue, though, is in order for the lysosomes to do their job and their enzymes to do their job, it requires an acidic environment. And so the inside of the lysosome is very acidic. So we want to make sure we keep those contents inside the lysosome and not out in the cytoplasm, major part of the cell. We also see peroxisomes. These are very similar to lysosomes. These are going to control things like free radicals that can potentially cause damage to our DNA. These are going to be really important in your kidney and liver cells because both of those are going to help filter out toxins, okay, at higher levels than other types of organs in your body. We also see that you have the mitochondria. The mitochondria is the bean-shaped structure. It's the powerhouse of the cell. This is like your electric company. It's going to do aerobic metabolism, and if you recall, aerobic metabolism means that it uses oxygen. It's going to use oxygen to break down glucose to make ATP. Remember, ATP is adenosine triphosphate, which is our currency for energy in our cells. The number in a given cell is going to vary depending on what the cell needs for energy. So some of your cells, like your skeletal muscles, are going to have tons of mitochondria, where other cells are not going to have as many because they don't need as much energy. These do, when we look at mitochondria, they do contain their own DNA and ribosomes in order to create more of themselves if needed. Now we have what we call the microtubules. The microtubules are going to function in maintaining your cell shape. A lot of the cells in your body, depending on their function, have a certain kind of shape, and the microtubules will help give them that shape. It also allows the cell for potential movement, not necessarily movement from like point A to point B, but movement of different parts in the cell and maybe on the surface of the cell. It does direct trafficking of substances within the cell. Again, it creates kind of like a highway or network, so things can move from one area to the other within the cell. We also see that microtubules are used to make cilia and flagella. The cilia is the hair-like extensions that are the real small ones, okay, that help move things across the surface of the cell, whereas flagella is going to be a longer extension that's going to whip back and forth for swimming. The only cell in our body as humans that have flagella is a sperm cell. And both of these, though, are going to aid in movement, whether it's movement of the cell or movement of substances across the cell. We also see microtubules make up something that we call centrioles. Centrioles are barrel-shaped bodies that are really important in chromosomal division. When we say chromosomal division, when your chromosomes and your cells need to divide, the chromosomes need to pull apart. And these centrioles are where they anchor those microtubules in order to pull apart those chromosomes. So they're really important in cell division. We also see that there's what we call microfilaments. These are thread-like structures. Microfilaments are really small, and again, these are going to help give a shape to the cell. Now, collectively, microtubules, centrioles, microfilaments, all of these things are considered what we call the cytoskeleton. They're the skeleton inside the cell, which helps give the cell shape. Just like your bones, your skeleton helps give you shape. All right, so now we're going to kind of switch gears a second. We've looked and reviewed the organelles. Now let's look at how the cell exchanges materials. How do things move in and out of the cell? And again, this should be review. So when we look at this, we first see that there's passive transport. Passive transport means no energy is required. No ATP is needed from the cell to do this job. An example of this is diffusion. Diffusion is the movement of a substance from an area of high concentration to an area of low concentration. If you'll recall, molecules like their space. Just like we have our kind of personal bubble and we like our space, so do molecules. So if they're crowded in one area, they want to spread out to a lower area until it's at equilibrium. And this is what diffusion is, all right? We also see osmosis is the diffusion, but it's the diffusion of water specifically. Water moving from high concentration to low concentration. So water gets its own name of diffusion, which we call osmosis. Now, osmosis does have some other terms that come into play. One is osmotic pressure. This is the tendency of water to move by osmosis. If there's more pressure behind it, it's going to allow for it to move. We also see when we put water with different substances, it creates a couple of types of solutions for us. And again, we talked about these back in biology and also in anatomy 1. We can have hypotonic solutions, and this is if a cell is placed in a hypotonic solution, water is going to leave and the cell is going to swell. And that's why I tell my students, if it's hypotonic, it's going to get really big. It's going to swell and then burst. And when it bursts, we call that lysis. On the other hand, you have hypertonic solutions where there's too much like salt or sugar in it, and it causes the cell to shrink or shrivel up, and this is called crenation. So in a hypotonic, the cell will get bigger, and a hypertonic, the cell will shrink. Now this is important. When you go in and you're dehydrated and they put an IV in you, they don't put pure water in you. If they put pure water into you, your cells would be in a hypotonic solution and they'd start to swell and burst. Okay? They also don't put in like a 10% salt solution. If they did that, you'd put your cells in a hypertonic solution, they'd shrivel up and shrink, and they can't do their job either. Okay, instead what they do is they give you a 0.9% NaCl solution, what we call saline. This is isotonic. This is the same type of chemical composition that your cells are in right now, and so it keeps them the same. So that's why it's really important that they don't just put pure water into you through an IV or huge salt concentration, because we want it to be in an isotonic solution, not hypo, not hyper. Another type of diffusion is called facilitated diffusion. Facilitated diffusion is the movement of substances from a high concentration from a low, but they need help. Okay, so not help from energy, but they need help through a protein. So remember we talked about how there's that barrier of the membrane. Some stuff can't move directly through it, and so they need help, and they'll use one of those proteins to move from one side to the other, still moving from high to low, but they needed help, and that's where the facilitated comes from. All right, so I do have some like kind of pictures or examples for you. If you look here in the first two pictures is simple diffusion. They can move from high to low across the membrane. But you'll notice over here in C and D, some things cannot just move across the membrane. They're either too big or they have charges, which they get repelled, and so they can't move through there freely, even though they want to do diffusion. So here's showing you again simple diffusion. Over here we have high concentration outside of the cell, and it's moving into the cell to spread out. So it's a simple diffusion. Okay, again, if it was facilitated, there would be a protein here which helps it move across. But since there's no protein to help it, this is simple diffusion. Now, passive transport means there was no energy required. This is like the lazy river at a water park. You get in and it just takes you around and you don't have to do any work. On the other hand, active transport means that you do have to do work. The cell has to do work. So active transport's the movement of a substance from an area of low concentration to an area of higher concentration. Now, this is against its gradient. So we talked about how molecules like to spread out. So what's happening here is all the molecules are over here and there's a few over here, and we're pushing them where they don't want to go. Okay, they're wanting to spread out, but we're pushing them all together. Okay, and so this requires energy to do this. So one good example in all of our cells is the sodium-potassium pump. If you'll recall, the sodium-potassium pump is sitting there pumping constantly. 3 sodiums out, 2 potassiums in, 3 sodiums out, 2 potassiums in, 3 sodiums out, 2 potassiums in. So it's pushing all the sodiums out of the cell and all the potassiums into the cell, and it's forcing them to go where they don't want to go, which means that it's pumping that, and that means it requires energy from the cell. We also see another type of active transport called endocytosis. Now guys, on an exam, please do not miss endo or exocytosis. The name tells you what's happening. Endocytosis is where things are going into the cell. Endo means in, cytosis means cell. All right, so substances are going into the cell. Now there are two types of endocytosis. One is called phagocytosis. This is cell eating. This is where the cell eats solid products, okay, and you can see that here in A, it's showing you endocytosis, but more specifically phagocytosis. This is what a lot of your white blood cells do for your immunity, which we'll talk a little later in the semester. On the other hand, you also have penocytosis. This is known as cell drinking, and this is where the cell takes in a lot of liquid with stuff that's dissolved in it, and you can see that here in the second picture where it's taking in a larger amount of substances that have been dissolved in that water. Okay. Now, on the other hand, you have exocytosis. Exocytosis is the release of materials from the cell. So they're exiting, they're leaving. So exo means exit or leave, cytosis means cell. So they're leaving the cell, which you can see here in this picture. All right. 2 more little topics that we're going to review before we get into the newer stuff in class. So this is energy production. Energy production, when we look at this, your energy is going to be obtained from 2 main things from your cells. The first is glucose, and that's the easiest, and that's what your cells are going to want to use. Now, if you recall, glucose, since it ends in ose, is a sugar. On the other hand, your cells can also use triglycerides, which are a fat. Now, these compounds, whether they're glucose or triglycerides, they're going to undergo a series of processes that we call cellular respiration. Now, this is where your cells are going to use oxygen in order to break things apart. Now, there are a series of steps that went with this. There was glycolysis, that's the breaking of the glucose into pieces. Then we enter into the mitochondria, where it goes through the Krebs cycle and the electron transport system. All three of these steps help produce large amounts of ATP. If we do all three steps in the cellular respiration sequence, we go from one glucose to creating 36 ATP molecules, 36 energies. But that's if we do all three steps. And so it's really important that oxygen's present and we have our mitochondria working to create that energy. Now, there are two separate and opposite phases when we look at how this energy is going to be used. There's what we call anabolism. Anabolism is where we're going to use the energy to create something, make something. And this is a metabolic process where we're making things. This results in the synthesis of complex molecules like proteins, like when you want to build muscle, or when you need to make new DNA, or when we want to store energy. So they're going to build things. That's anabolism. On the other hand, you have catabolism or catabolism. This is where energy is going to get released when you need it. This is the breakdown of your nutrient sources. So like when you eat stuff, your digestive system is going to break apart the food you eat into its pieces, releasing energy in the process. Okay, that energy then can be used to build things in your body, which then we can release and then we can build. It's a constant cycle between anabolism and catabolism in your body. The last thing we want to talk about is reproduction and differentiation. Now, when we look at reproduction, we have two types of reproduction that happen in our body, okay? We have asexual and we have sexual reproduction. So we're going to focus mostly on, in this class, the asexual reproduction, like when one cell is going to divide into two, and that's going to be more for healing and things like that and repair. On the other hand, sexual reproduction is where we create gametes so that we can create new human organisms, okay? So proliferation is a regulated process by which cells divide and reproduce, and they can divide in two ways. Mitosis is the asexual form where one cell just divides into two. So one cell undergoes one division to make two cells. So we have one cell divides into two. Okay. Now, if you recall, mitosis has a number of phases. There's prophase, metaphase, anaphase, and telophase. I've always taught my students to use their hands to demonstrate these phases. Okay, so prophase is where the cell is getting ready to divide. The chromosomes are condensing. Okay, so I can actually see them. My fingers are the chromosomes, and the nucleus starts to disappear. In metaphase, all these chromosomes line up in the middle. Okay, they're lined up in the middle of the cell. In anaphase, the chromosomes start to pull apart because the cell is dividing into two, until in telophase, I finally have two new cells. All right, so that's kind of the progression: prophase, metaphase, anaphase, telophase. In meiosis, it's a little bit different. Meiosis is where one cell undergoes two divisions to create four cells. Now, what's unique about this is in meiosis, we're going to create all unique haploid cells. Haploid means they have half the chromosomes. So for us as humans, we have 46 chromosomes. So when we make an egg or a sperm, we want them to each have 23. So that when one egg and one sperm fuse together, we restore it back to 46. All right, so meiosis is going to be used for sexual reproduction, and that's going to be talked about more in Patho too when you get to the reproductive system issues. Now, differentiation is when these cells have undergone that process of dividing and now have two cells. Now they need to have a certain kind of job. Okay, differentiation is where the cells are given a specialization. So the cell type is going to reflect its function, which is going to reflect its structure, and it's going to do its particular job. All right, and so a heart cell can't decide it wants to go be a liver cell because it's been differentiated. It not only can do that heart cell job. All right, so it's really important that those cells differentiate. In cancer cells, what happens is they lose their differentiation. That liver cell that should be doing a liver job, it decides it doesn't want to. Now, it doesn't become a heart or a kidney cell or anything like that. It just becomes a cancer cell where it's just worried about itself and it just keeps dividing. So it'll do proliferation, but it won't do differentiation, which causes it to be a tumor, which is useless, just taking up space, and then starts invading neighboring areas. All right, and so it's really important that proliferation and differentiation work together. Right, so in your textbook you have an example here. This one shows you mitosis, one cell dividing into two. Over here you have meiosis. It goes through two divisions where one cell ultimately divides into four. Now if you'll notice in mitosis, all the cells should be identical to the original, whereas in meiosis they have half the chromosomes and they're all unique. All right, so there is that difference, and you can review that and look at more detail in your textbook, but also you can review it in like your anatomy book or biology notes, things like that. All right, and so that is where we're going to leave off in chapter 1. We will be starting from this point on in lecture, where we're going to dive into more of the patho part. Now when the cells are doing things that are different, we're going to look a little bit at genetics and even cancer in this first chapter. All right, so again, make sure that you've reviewed this, watched this before the first face-to-face class.